Okay, I'm redoing this lecture for you. This is actually lecture 3, week 1, looking at descriptive statistics. Unfortunately at Gatton we had a power cut right at the end, and so I don't think Echo 360 recorded this, unfortunately. So what I'll try and do is skim through it so that you all can have this lecture. What I'm going to talk about in this lecture is biometry as a toolbox, which I actually talked about in the first lecture really. But remember how useful biometry is to give us some tools at our disposal on how to cope with data, how to think about research questions, how to analyse, set things up, save, interpret and so on. And one of the things we might look at first is biological variability, because unlike other aspects like maybe manufacturing sort of quality control and so on, in biology, when we're looking at biological experiments or agricultural experiments, we've got that added factor of the biological variability of our subjects. So whether you're thinking about an animal, a plant, a soil sample, trees, whatever your unit is, or even your wildlife studies, we've got that biological variability. And I like to think of it as 3 aspects of biological variability. You've got your genetic variability, you've got the environment effect on that genetic variability, and in agriculture I'd like to think of the M as well, which is how we manage our experiment, or how we manage the biological subjects. For instance, you know, we have got the genetic side of, yes, we've got this this breed of sheep, we're using a particular breed, particular genetic background. Then we've got the environment, that's how we look after them. Are they extensively grazed? Have we got them in pens? What aspect are we looking at? And then also we've got that management side, like, you know, how often are you feeding them? What are you feeding them? What disease control are we using? So you can see that any aspect, whether you're going plants, animals, soils, you've got that biological variability. Look at data and data types, look at some data and some of the measures that you should all be completely, hopefully familiar with. At least 95% of you, I should think, would know about the central tendency and the spread, 5-number summary plot and the use of box plots. And then hopefully I'll get into talking about some R&R Studio and descriptive statistics. Since I'm redoing this, I might do that as a separate add-on for you, and look at interpretation. So our aim in this course is to give you the basics for statistical reasoning, for being able to look at applied statistical design of experiments, so you can understand how they're designed and why, and use applied statistical data analysis, which links very closely to the design of experiments. And after this course you should be equipped to design a simple randomized experiment yourself and understand the principles of design, to conduct the analysis of simple controlled experiments, and to continue learning more about advanced statistics. If you want to continue more, next year there will be a second statistics course in advanced statistics for those of you that might go into research or enjoy statistical analysis. First of all, let's just think back to biological variability, and I alluded to just now about the fact that manufacturing or some of the industrial processes might expect things to not have that much variability. Obviously these knives look pretty similar, and the only thing you might notice is, you know, they might have got some specks of dust in the mix, or there might be a slight variation in colour, there might be a slight variation in weight. Basically, you know, one knife is like the other. However, thinking about biological variability, and I've just put some fungi up here as an example, but obviously if you look at animals, plants, fungus, all sorts of biological organisms, there's many aspects to their variability. One is the genetic, what breed is it male or female, that type of thing. Is it from a elite breeding stock, or is it a new isoline in our crops? Is it a particular variety? Is it a subtropical variety? Got all those aspects. We've also got the environmental aspects on things. If you've got too little water, not enough nitrogen, you're going to have a lot weaker and lower yield than if you've got adequate environmental conditions. And then I like to think about the management side, which is what you do as a experimenter or a farmer or a production person in agriculture to try and maximize your yields or your outputs. So let's just look at these numbers. What can you say about them? Not much, maybe. Exactly. We don't know much about them. We just know that we could probably find a minimum and a maximum, but we've got no idea about this data. Is it useful? Might give you a bit more information here. We've actually got 10 samples. So here, we've at least got some idea that we've got 10 samples, and then we've got several numbers within each sample. But even that is not very useful. Data needs context. So the information you need to find out what is being measured. How was it collected? When was it collected? Who collected the data? Was it collected in one day? Are there any biases in the data? What are the units? And these are all relevant. For instance, if you look at how was it collected, if it was collected by school children in one of their science classes, the quality and the understanding of the science would be very different than if you had someone with a degree or working on a research program who were looking at very strict protocols on how they collect the data. When it was collected can be important. Usually you want all your data to be collected sort of in a one day. You want it to be collected by people that know what they're doing. You want to know if there are any biases in the data. If I was looking at, for instance, I wanted to know the height, weight, the chest size of young men because I was going to go into making tops for them or something for rugby, whatever, you would have to try and get a representative sample. You wouldn't take maybe the basketball team if you're looking for measurements for rugby and that type of thing. You've got to think of what the units are, and typically these days we use SI units. But one of the things you can do in your particular study is look in the literature, read a couple of papers and find out what they use. I mean, if you're looking at weight or yield, what are they using? Kilograms, grams? Are they looking at yield per acre, yield per hectare, yield per metre squared? All of those things become important in your study. Let's just now look at data types. The main comparisons you can think of is the continuous variable versus the categorical. And we'll just look at this. In class I did ask people to shout out a few examples, but we'll just go through these fairly quickly. Interval data. An example of an interval data is something like years. It appears to be continuous data. You can have year 300 AD, go up to 2014. But they are sort of a ranking in a sense, and the value really doesn't have a clear zero. They are useful in some ways, but one of the key continuous variables that we come across in biology is the ratio variable. And this is something like length, weight, height, can be salinity, all sorts of measures where you've got that continuous measurement and it does have a value. It does have a meaning such that if you have zero weight, there's nothing there, and so on. Let's think about categorical now. There's the type of variable that's a discrete number. You've got the number of plants. If you've got bacterial counts, you can't have half a bacteria. You look under the slide, under the microscope, and you count the number of bacteria. A discrete variable can only take integer values, so you're either going to have 6 pregnant ewes or 5 pregnant ewes. You're not going to have 5.5 pregnant ewes, that type of thing. You've got the nominal type of categorical variables, such as colours or the sex, male, female, the model of tractor, the breed of sheep. What you're thinking of here is N for name, if you like, where something like a red and a blue, one's not bigger than the other. There's no order. And so moving on from that, you can imagine that we have got the ordinal type of categorical variables where there is an order implied. A typical example is a shoe size, hat size, even a disease rating, where it's not a continuous variable, but you've got some sort of rating, so that a 1 might be a lot less than a 5, and so on. Also, in scales where you're doing survey data, you might be asking people's opinions. There's a thing called a Likert scale, and that might be a typical 1 to 5 or a 1 to 10 rating, where you're asking people to give a rating, and again, that's an ordinal variable. Just thinking of agriculture though, if you yourself was asked to quantify senescence of leaves, for instance, this is just some oak leaves. You can see I've got 5 leaves. The one on the left here is fully green. The one on the right is fully senesced, which means it's dead. And in between, I've tried to get a range of different senescences. You know, the one in the middle is supposed to be about, you know, half senesced. And the second one in is just starting, and the fourth one in is getting close to being fully senesced, but it's not quite as dried. So you can see you could give that a rating of 1 to 5, and you could use that in your study. Let's just have a quick think about what descriptive statistics we do have. And when we think about the measures of central tendency, what we're saying is we want to summarise our data with one value. And typically we can think of the mean, which is also called the arithmetic mean, the median, which is when you order the variables in order and then take the middle value, or the 50th percentile value, okay? And then the mode, which, if you know anything about French, à la mode means fashionable or something like that. You can have ice cream, or dessert à la mode often means you're having ice cream and it's really fashionable to have something with ice cream. So just think of mode as, you know, something to do with fashion. What's popular? It's the most frequent value. So if you've got a whole load of data and you're asking make of tractor, if you've got more Massey Fergusons than Fords, then that's your modal type of data. Measures of spread. These are more related really to the way we look at data with the mean and so on, and I'm going to talk about these. The variance is something you'll hear about, and we'll talk about that shortly. And then the standard deviation. The standard deviation is the square root of the variance and brings us back to that same level of magnitude as our mean. So if we've got, let's say we've got mean milk yield might be in litres, so our variance will be in litres squared, and our standard deviation will be back down to litres, and I hope that'll become clear as we go through. Another thing you might notice for measures of spread, you can always take the maximum minus the minimum. That's called the range. And you've got the interquartile range, which is when you take the 75th percentile taking off the 25th percentile, so you've got the middle 50% of the data, and that's called the interquartile range. I'm just going to mention very briefly here about validating and valuing your data. When you collect your own data, you really need to consider and make this validation step after you've collected it. You need to look for mistakes, you need to look for typos, and if you're transferring data from a clipboard to a database on a computer, you need to think about whether you've got any mistakes, any outliers. Outliers might be something when it's a magnitude of difference. Instead of putting 50 in, you put 500 in. That could be a typo. And likewise, you could have a lot of zeros. It could be that you've got faulty equipment. And these are things to consider and comment on in your data collection. The other thing I'll mention is Excel is a very useful spreadsheet tool, often useful for setting up data sheets for collecting data. It's useful for putting data in or collecting data into the computer, but it's not a statistical package. It's very useful, and I would expect that most of you, if you're going to move into science, would have to have good Excel skills, and I hope that during this course you'll have to give yourself the chance to improve your Excel skills and help each other with those. It's very useful for storing and backing up data, and I'd say Excel is essential in most sciences. One of the things you need to learn early on in this course, and we'll be doing it in some of the workshops, or even I'll show you in lectures, is to convert your Excel file into a CSV file. And just to note here, those that you don't know what CSV stands for, it's comma separated value. It's a nice simple flat file that allows us to take that Excel file into our software, whether it be R or other statistical packages. The other thing to note is that when you have any files on your computer, make sure you back everything up, and also keep things at a second site or on a stick for safety. We don't want any lost data. So when you're looking at a research study, you start with a research question. Typically, once you've designed your research question, you collect some data, and I hope after you've done this course you'll have a really well designed study, and you'll have all the skills that you might need to set yourself up in designing studies. You may have collected some data such as a continuous variable or a categorical variable. For instance, we've already talked about these. You could be collecting yield, which is continuous, or disease rating, which might be categorical. You wish to summarize the results. We wish to find out very often whether there are differences between our treatments. We might have got different varieties, different breeds, different management practices. We need to find out what's different, or whether they are or are not different. We wish to put some measure on the reliability of the estimates, and so we used appropriate statistical testing and also statistical reliability scores such as confidence intervals to put some reliability on our measurements. Just showing you very quickly here, I hope that you already are very familiar with this. We can see the mean here, and I did ask the class if they could describe how to collect a mean, and correctly a couple of students mentioned that you add up all the variables and divide by the number of variables. So that's what that X bar at the top there is showing you. X bar here, and that is X with a little line across. That's what we use as our notation for our sample mean, and all we're doing here is notating the fact that we're adding up the x1 right up to xn, whatever that might be. If we've got 10 in our sample, then xn would be x equals 10. So we're adding up those variables, dividing by n. And in this equation here, this is just taking out the 1 over n to the front. So this is coming out to the front, and we're just saying in our notation here, sigma, which is add up or sum all of these xi's, where the I is equal to 1 up to n. So as I said, if this was 1 to 10, our xi would be I equals 1 up to n, which is 10. And these notes, if any of you are buying the Ploughing Through Biometry course reader, they're all in those notes for you. Let's just look at what a deviation from the mean is, and I'll just explain this to you. So here we have the variable. It doesn't really matter what it is in this case. We've got variables starting from 10 up to 25, or individual values start up from 10 to 25. And these red dots represent all the data points, okay? So these are just all our data points, including this one up here. What I've got here is a line across. This is for this particular sample, that's the mean. Mean is 14.7, okay? And here's a little mark showing you the mean on this vertical display of data. You can also display this horizontally, but just for now, let's see this as above the line and below the line, which may be helpful. Okay, so now let's look at the deviations from the mean. So if we take this point here, this black arrow shows you the distance that that point deviates or is different from the mean. Okay, so that's quite a large deviation, and it's a positive deviation. It's a lot bigger. It's something like 24 or 23 something. Here we've got some points. So this point here is a deviation below the mean. Here's another point, deviation below the mean. So each individual value has a deviation from the mean. Obviously you might get 1 or 2 on the mean, but anyway, this is for you to understand. It helps us later when we're trying to calculate variance, and there are 2 calculating methods for calculating variance by hand. You can see my notes, and you should be able to calculate the variance with your calculator, and I'd like you to do it also in Excel. If you learn to input the values into your calculator, that will be very useful for you in the future. So here is what our sample variance looks like, and here this is what we call sample variance, S squared, S superscript 2. And so let me just go back for a minute. Here we're talking about deviations from the mean, and the reason I wanted to put that first is because this is a deviation from the mean. x1 minus x bar, that's a deviation from the mean, that's a deviation from the mean, and so on. Now you might wonder why we're squaring them, and I hope that if you think about this, and I'll show you another way of looking at this, if you added up all these positive deviations and then added up all these negative deviations and put them all together, it would sum to 0, because you've got as many above as you have below. And so if you summed up something and you want to get an average deviation, it's not very useful to sum them all up and they're equal to 0, because you can't get an average. However, if you look at the variance formula, it takes those deviations and squares them. So whether they're negative or positive, you know that a squared number a squared negative number is a positive number. So this makes all our deviations positive, okay? And then we just divide by n minus 1, which we'll discuss probably later. Sorry. I just want to show you that in Excel, you can work out these things. You can use the formula average. And I hope that most of you will know that this here is a column address. It says take B4 through B13 and get the average. The standard deviation, you can do SD.S of that those same values, and you can get the variance of the sample. So var.s gives you the variance of those. So that's just I would like you to try and learn how to do this in Excel as well, just be useful for you in the future. This is the calculating formula. Now I'm not really going to, it is in the notes if you do want them, but you won't really need to necessarily be able to use you won't need to do this for anything, but it's there just for completeness. This is the variance, and there's two ways that you can actually calculate it by hand. And these calculations were produced before we had calculators, really. So when you had to do everything by hand, this was a quicker way of getting this. Nowadays we can use Excel, we can use calculators. Let's just again look at the standard deviation. It's the square root of the variance, and since the variance is in squared terms, it brings the measurement back to the same scale as the original data, and it represents the average deviation of values in your sample from the sample mean. So what we're doing here, I've just showed you before how to do the variance in Excel. You can do this in Excel. But the other thing is that S, our standard deviation, is the square root of our variance, S squared. So that's something that you should get very familiar with. So let's show you some data, and I'm going to show you in Excel just so that you get this as familiar. We've got some items here. These are people, in fact, and then we have X, which is our variable of days absence from work. They're in order here, so we've got a couple of people who weren't absent in the 6-month period, and we've got person 10 who has 28 days off in a 6-month period. So that's our data. Doesn't really matter what it is, but I just want to show you how you can use Excel. So what I've done here is I've popped that data in here. I've transferred it into another worksheet, and then I've got the deviations. And I hope here, you can see here, I've called it deviation. I'm saying that this is our X, which is, these are our X values, and I'm taking off the mean. Let's just pop down here. The mean is 8.3 for this data, and I've used that formula, 8.3, which I hope you can all do in Excel. If you can't, then we can help you on discussion board with that. So here's X minus X bar, and so here we've got 0 minus our mean, minus 8.3 gives us minus 8.3. And we go through and we do that for all of them. And as I said before, if you add these up, it will come to 0. And that's what I think is useful, because if you understand what these deviations are, that's quite useful for your understanding of the variance, because here we've got the squared deviations of the mean. So here we've got these values here, minus 8.3 squared. As I said before, everything you do here becomes positive. So as soon as you square a negative number, it becomes positive. So all of these negative numbers are now positive. And that means instead of adding to 0, if we add up these, which is down here, sigma x bar squared, x minus x bar all squared, that's our value. So that's our variant, our sum of x minus x bar. And then to get the variance, you divide that by n minus 1. And then to get the sample standard deviation, you take the square root of that. There's also a second method, and this is just shown for completeness, and you can have a look at this in an Excel spreadsheet that I'll put up on Blackboard for you. Yep, there's also some deviations. If you want to have a look at this, it's actually in a little screencast that I made on Vimeo, so you can have a look at that if you want to take a little bit more time on that. And this is where we're up to for week 1. So I'll stop now.